Before jumping into the details about how beta-carotene is metabolized within living organisms, I want to show this quick schematic which gives a summary of what's going on. So essentially we have rhodopsin, which is able to absorb light, but rhodopsin is a complex that is made up of opsin and 11-cis retinal. So essentially what happens is that we have light absorption on rhodopsin, and that light absorption, it leads to excitation, because essentially what happens is that when this light is absorbed by rhodopsin, a photoisomerization event occurs in which we produce all trans retinal. But in this photoisomerization process, rhodopsin is able to interact with transducin, which is a G protein. And by interacting with the G protein is able to prevent calcium 2+ from entering the rod cells of the retina, which further causes that excitation in a signal transduction pathway. Now, when we have all trans retinal, we have to convert it back into 11-cis retinal in order for that excitation and light absorption to occur again. So this is somewhat of a cycle. And in order to go from all trans retinal back to 11-cis retinal, there are three steps that we must take: a reduction, isomerization, and an oxidation reaction, which we will now go into detail about. So over here we can see the structure of beta-carotene, and essentially what happens is that the beta-carotene is cleaved in half and we get two products. We get all trans retinal and all trans retinol. Now, what's important to note over here is that we are interested in all trans retinol because this all trans retinol, it can either be converted into another product or it can be converted into all trans retinol, and we can utilize all trans retinol to undergo the further changes to be utilized for that nerve excitation. So the important thing you need to know over here is that beta-carotene will cleave in half, and these are our two products. Now over here, we can see that, first of all, as I mentioned, we are interested in retinol. So one of our products, it's a retinal. We have all trans retinal, and we're going to convert that product into all trans retinol. So now we're going to have two products of all trans retinol, and both those molecules are going to undergo that same pathway now. So we have two all trans retinols, and the enzyme that catalyzes this reaction when we're going from all trans retinal to all trans retinol is retinal reductase. So retinal reductase, and as it's in the name, reductase, so it's utilizing NADH to gain electrons. So we're going to see NAD+ as one of our products because we're utilizing NADH. So now we've converted that product that we didn't want as retinal, and we've converted it into retinol through the enzyme retinal reductase. Now we have two all trans retinol molecules, and we're going to convert them into 11-cis retinol. Now, in order to go from all trans retinol to 11-cis retinol, we're going to utilize the enzyme known as Retinol Isomerase. Now, it's in the name, so it's a retinol, so the enzyme is going to be a retinol, and we're undergoing an isomerization reaction where we're simply going from trans to cis. So retinol isomerase. Now, once we have 11-cis retinol, we want to convert it into 11-cis retinal. Now, going from this molecule to this molecule, we're undergoing we're going to need another type of reductase enzyme. So this time we are going to be producing NADH, and we're going to be using that NAD+. So this time it's an oxidation reaction, and it's going to be retinal reductase once again. Reductase. Okay, So now we're at 11-cis retinal, where it's undergone an oxidation reaction and it's lost electrons, and those electrons have been picked up by NADH. Now we have 11-cis retinal, and we are interested in 11-cis retinal because remember that rhodopsin is a complex that consists of opsin and 11-cis retinal. So now this 11-cis retinal and opsin complex, known as rhodopsin, is now going to absorb light. When it absorbs light, the 11-cis retinal is going to be converted into all-trans retinal. So to go from 11-cis retinal to all-trans retinal, all we needed was the input of some light. Now we can see that we're back to all-trans retinal, and we're going to see up here, it's the same pathway again. So first we're undergoing a reduction, then an isomerization, then an oxidation, and then simply light energy will convert it to all-trans retinal. Now, we'll look at the structures next, but to quickly overview, remember that what are we interested in? We are interested, when we cleave beta carotene, we have a retinal compound and we have a retinol compound, and that retinol compound, we're going to convert it into retinol, so we're going to end up with two all-trans retinols. They're going to undergo this pathway once we get to 11-cis retinal, and then we're able to have rhodopsin, because rhodopsin is a complex with 11-cis retinal and opsin. It's going to absorb light, which is going to lead to nerve excitation, and we get all-trans retinal as a result. Now over here we can actually take a look at the structures. So we have all-trans retinal and all-trans retinol. These are the products of the cleavage of beta carotene. Now remember, we're interested in all-trans retinol. So this all-trans retinal compound that is also produced by the cleavage of beta carotene, it will be converted into all-trans retinol through the enzyme Retinal Reductase, and this is actually an oxidation step, because it's a reduction step because NADH is utilized and we will be gaining electrons. Now this all-trans retinal, it can also be converted into this, but we are not interested in this compound right now. We're just talking about it being converted into all-trans retinol. Now we have the two all-trans retinols, and they've been reduced with the help of NADH. Now remember that this all-trans retinol will actually be converted first into 11-cis retinol, with an O, retinol, through an isomerization reaction. After that isomerization reaction, the 11-cis retinol is converted into 11-cis retinal. Now 11-cis retinal is what we are interested in. And to go from 11-cis retinol to 11-cis retinal, it's an oxidation reaction, so we'll actually be producing NADH going from here to here. Now, 11-cis retinal, that's what we are interested in. So this is the component, the visual pigment of rhodopsin. And once we see that light absorption, that visible light absorption, we get all-trans retinal. But in that process, during that process of when light is being absorbed on rhodopsin, it leads to a photoisomerization event, which leads to all-trans retinal. But this isomerization reaction actually allows rhodopsin to interact with a G protein known as transducin. And transducin is the protein that will prevent the entry of calcium 2+ into the rod cells of the retina, which will inhibit the exocytosis of neurotransmitters. And through further complex pathways, we will see the excitation and signal transmission to the brain. So, and that all-trans retinal that is produced, it will once again just start from the beginning and undergo that same reaction again. And the end result of this entire pathway is a neural signal which allows vision. Now, to quickly show that schematic again, we have all-trans retinal, but remember, we're interested in all-trans retinol. So it's going to undergo a reduction pathway where we will utilize NADH to get all-trans retinol. that all trans retinol will undergo an isomerization reaction to produce 11-cis retinol. Now, 11-cis retinol will then be oxidized, so we'll be producing NADH, and the 11-cis retinol will be converted into 11-cis retinal. Now, when 11-cis retinal and opsin come together, we actually have rhodopsin, and this is the visual pigment of rhodopsin. And once we see that visible light energy absorbing, we then get the end result, which is excitation and excitation to the brain and the signal transmission. And that is essentially the metabolism of beta carotene. It's just a cycle of steps where we want 11-cis retinal, and we want that complex of rhodopsin to absorb light to allow that nerve excitation.